So for the people online, you will not be able to ask a question in the chat, although you will be able to unmute yourself and ask question if you have any question at the end of the presentation. And today's speaker is Masoud Ghaderi Zefreh. So from the Roslin Institute, he is from Iran. His background is in engineering and mathematics. Masoud joined the Roslin in 2019 to work on a European project called Small Ruminants Efficiency and Resilience, and he is currently studying effect of selection for novel phenotypes on the biology of animals. Masoud, the floor is yours.
Thank you very much. Hello, everyone. So this is part of the Smarter project that Clemens mentioned. It's a European Union Horizon 2020. So as we all know, animals are exposed to a variety of challenges throughout their lifetime. And this can be, is there any way I could get rid of this bar below? Yeah, probably not. Never mind. It's okay.
Sure, stop the presentation and start again. Yeah.
So these environmental challenges can be anything from shortage of food or a dirty environment or pathogen existence and other factors. And it is expected that in general, the more the environmental challenges are, the lower the performance of the animal is. But the level at which the performance of the animal changes in the presence of environmental challenge is important, and we call that the resilience. So assuming that there is a linear relationship between the performance of the individual and the environmental challenge it is exposed to, we can define the resilience to be the slope of this line and the production potential to be the intercept of that line. So basically that way we can assign a line for each individual, and to estimate these lines means that to find the coefficients for this line, which is the production potential and the resilience for each individual. This is possible using random regression models where we estimate basically breeding values for these two coefficients for each individual, assuming that the environmental challenges are known. Now, there are two problems. One is we seldom know the environmental challenge level for each individual, which means that this has to be estimated itself, which we can do so by using reactional models, where we estimate all these three components. Another problem is that often we have only one data for each individual. For example, we have the carcass weight of the individual after it dies. So estimating a line with one record for each individual means that we need to use the information from other related individuals. And these two factors can decrease the accuracy. Now, this is where genomic prediction can come to help to increase the accuracy for these breeding values. So the objective of this study was to first look at the benefit that we can get from using genomic prediction in random regression or reactional models, and also estimating the effect of other factors on the accuracy and on the bias of breeding values, namely the thing that I mentioned earlier, the uncertainty in the challenge level, and also other things we can do like phenotyping strategies. So this was done in one study, and in the second study, we assessed the response to selection in different selection strategies, and everything was done using simulation. So for that, we simulated the genotype of sheep, a sheep population. So not taking into account the sex chromosome, we had 26 chromosomes, 500 QTL for each of the traits, resilience and production potential, and 1,000 SNPs for doing the genomic prediction. And we tuned the parameters such that the mutation rate and so on, such that the population had an LD structure that was close to the real data. And for the first study that I mentioned earlier, we had only 3 generations of individuals, so without selection, just looking at the accuracy and the bias of EBV. In the second study, where we wanted to do the selection, we had a different pedigree structure with 10 generations and selection in between each generation. And in both of them, we had large half-sieves, so like families that are mainly half-sieves, but also some full sieves, just to make imitate a realistic scenario for sheep population. For the phenotype, as I mentioned earlier, we are assuming a linear relationship between the performance and the challenge level, so that is y and x. The other two coefficients here are assumed to be heritable, so having a genetic and environmental contribution to these two effects. I tested for different heritabilities, but I'm going to show only the low heritability and the negative genetic correlation between resilience and production potential, because this is the worst case scenario in terms of the accuracy we get in general. So after we can now here simulate the values for production potential and for resilience. For challenge level simulation, we need to define basically the range of environment. And that is mainly the interval at which we have our data. We can have a large interval, basically, which is a wide environmental challenge that covers a variety of environmental conditions. And 0 to 2 here I chose because this is 6 standard phenotypic deviation, or narrow range of environment, either covering only areas that are typically good or environments that are pretty bad. And then once we define the whole environmental range, now we can divide it into subsets, which we call herd. So herds are where basically individuals live in. And this herd covers only a percentage of the whole environmental range. So if I take, for example, this scenario, I can have herds that cover only 10%, 20%, and 30%. And that I call the herd heterogeneity. Now, the challenge level for each individual, which will be simulated, is defined by the herd that individual lives in, because individuals in the same herd in reality are exposed to more or less similar challenge level. And again, 3 different scenarios was considered. So the random scenarios, siblings live in different herds. So a family members can be living in different herds and exposed to different environmental challenge levels. In the cluster scenarios, families live together. And in the assortative scenarios, not only they live together, but there is also a correlation between the challenge level that a family experiences and the overall performance of the family in terms of their sire. Finally, for the selection scenario, we consider 2 cases, one using a selection index and the estimated breeding values for resilience, R, and the production potential, 0. The index here, B, is basically the weight for resilience and was allowed to vary between 0 and 1. And a case where we do not do reaction norm. So assuming that we use a conventional model without actually affecting for reaction like resilience and production potential, only affecting for the herd effect and assigning a breeding value for each individual, a general breeding value for each individual. Okay, so now looking at some results, what you see here on the left is the accuracy of EBV for production potential and resilience in 3 different scenarios. So again, random is when family members do not live together. Clustered is when family members live together. And assortative is the cluster scenario where there is also a correlation between how good the environment is and how good the sire is. For example, best families live in the best farm and the worst families live in the worst farm. And colors here represent different methods. So I have G-blob red and the other one. I'm using a pedigree-based model only with the other color. So what we see here first is that, yes, there is a massive increase in the accuracy of breeding values up to a factor of 2.5. In general, production potential is better estimated for resilience, but the bad news is that the resilience accuracy does not change in the way we put individuals in the farm. So there's nothing we can do to improve the accuracy of resilience. However, the production potential is very sensitive to the way individuals are allocated in different scenarios, and with random scenario having the best accuracy and the assortative having the lowest accuracy. The result for the bias were the same, so I excluded the figure for that. There was again no improvement or decrease in the bias for resilience. Production potential was sensitive and followed the same trend. So this plot shows how the uncertainty in the challenge level affects the accuracy. So as I said earlier, when we do not know the challenge level, we have to estimate it. And this is normally done by estimating an overall challenge level for all the individuals that live in the same herd. So here RN10, it represents the case where we have herds covering 10% of the environment, this schematic figure, 20% and 30%. And RR is the random regression analysis, a hypothetical scenario where we know exactly how much each individual is exposed, like how much the challenge level is for each individual. So what are we seeing here is that the more heterogeneous the farms are, so the more the extent of this farm, the lower the accuracy for production potential and resilience. And again, the best accuracy for within each of these scenarios for production potential is the random scenario, and the assortative has the lowest accuracy. Again, even within different analysis, there is not much we can do to improve significantly the accuracy for resilience. However, the good news is, even though we do not know how much each individual, how much the challenge level for each individual is, still we get an accuracy using GBLOG that is around 30%, so not too catastrophic. And this is my last slide on the results section. So we're looking at the gain or loss that we obtain after 10 generations of selection using different strategies. So remember that these figures are wrong. They should somehow, they are not represent, but this is wide range of environment. So if I go back, a wide range of environment is where we have a variety of a very large interval that covers a variety of environments, and then I'm looking at the other scenarios. So this figure is when we are simulating our data in a wide range of environments. And on the x-axis, we have the selection index for resilience, and on the y-axis is the gain. So what it means is that if we totally select for resilience, one means pure resilience. Of course, we are improving the resilience, but because we have a negative genetic correlation, we have a loss in the production potential. And the other extreme shows the other opposite. And because we are covering a wide range of environment, we have this nice X shape where at some point we can improve both of them. What is also more interesting is that if we use the conventional model without taking into effect the reaction norm and all this complicated model, we can still improve both resilience and production potential at the same time, and that is shown by these triangles. The same colors and shapes are used in the other figures. So triangle is when we use the conventional model without reaction norm. But what is happening is that now we are looking only at a narrow range of environments. And you see this X is becoming a little bit distorted. And eventually here, when we are looking at very bad environments, selection results on only improving the resilience and not production potential. Of course, we can change the index and kind of limit the loss we get from after 10 generations of selection. In the other case, where we are only selecting individuals in good environment, we are most of the times only improving the production potential and not the resilience. So in overall, the genomic prediction improves the accuracy for resilience and production potential, and it's a great tool that should be used. The uncertainty or unknown challenge level decreases the accuracy, but even though the accuracy is decreased, it's still in a range that can be used for selection. The estimated EBVs can be used for selection. The distribution of animals across environments, both and the way we collect phenotypes from animals, both affects the accuracy of the breeding values and also the response that we get from the selection after 10 generations. So thank you very much. This is the first the first talk I have. I also have another one, which another tool that I want to present after this one. If you have any questions, I'll be happy to answer before I go to my second talk. Thank you very much.
So we get a question for Masoud for the first talk now.
Yeah, Masoud, I have a question, yes, that I didn't get in a previous slide. Please. Index for resilience, this is the, what is this number, the regression coefficient?
So no, this is actually B. So assuming that I have estimated a breeding value for resilience and a breeding value for production potential, if B is 0, I'm basically selecting only for production potential, okay. And if it's 1, I'm basically selecting for.
So this axis is the regression coefficients of the index, right?
This is the weight of the index.
The weight of the
yeah.
Okay.
So I guess you haven't simulated that. I wanted to know what is your opinion, because this assumes a linear relationship. But what happens if you don't have linear relationships? You could, for example, fit a multiple line model in which you could estimate slopes for each one of those segmented lines, right?
Yeah.
But then you would have to take care of the correlation between the slopes at each one of the segmented lines. What would be your guess? How would you think that the results would change?
Yeah. So what would be before I think about it, like why would we need to have different segments? Is it like
you have a curve, for example, an S shape rather than a line?
Yeah.
Or something even more complicated. You could segment that line into individual lines and then estimate the slopes at each one of those lines for the trajectory.
Okay, I'm just moving a little bit because I know there is a problem with the audio. So can someone from the people online?
So Julian said that they hear us.
Okay, great. So having one line has this benefit that we can actually take the slope and call it resilience. If you go for like more nonlinear functions, then defining the resilience based on the coefficients becomes a little bit challenging. Now, that's a good question. That what will happen if you have different lines? I guess how you would define the resilience is by collective of these slopes, right? So basically, individuals will have different resilience on different locations. I haven't done this analysis, to be honest. It sounds like a good idea. But this way, yeah, we also need to allow for different correlations with different environments as well. Yeah. So I do not know how it will change the result. Basically, if you get positive genetic correlations, I didn't show, but everything becomes fantastic. Even not knowing the environmental challenge does not affect anything. So I guess if we do that, it probably improves the results because we can allow for 0 and positive genetic correlation.
And here's another question. When you compute when you put the formula, the production potential plus environmental challenge multiplied by the resilience. And the problem is that you need to estimate that environmental challenge. Yeah. Do you then estimate a single value for the population?
No. So what happens is that here, when I simulate, I actually go back to this because this was better. So when I'm simulating the data, I record three things. One is the ID of the individual, four things. One, the ID of the individual, the herd it belongs to, the exact challenge level, and the phenotype, right? Now, if I assume that I do not know the challenge level, I end up with the ID of the individual, the herd it belongs to, and the phenotype. Now, for all the herds, I will estimate, for all the individual within a herd, I estimate a challenge level. And that is basically the scaled average phenotype. So individuals that have, so a herd that has an average, a higher average of performance means that all the individuals in that herd have lower challenge level. In another herd where the average performance is lower, means that the challenge level is higher. So I take these herd averages and define the challenge level based on the herd average.
So all the animals within a herd have the same.
Exactly. So all the animals within the herd that would have the same challenge level.
And maybe if you could put a different from this, it will change like because random, cluster, and associative will have less effect if you are able to adjust that regression coefficient to each of the animals.
What do you mean the regression coefficient?
This regression coefficient that you use to estimate the two traits?
The X value, yes. So yeah, this is different. It's a different analysis. Either you change the herd extent, being 10%, 20%, and 30%, and that affects the accuracy in a different way, or you define like how within a herd individuals are distributed. Like, do families live together, don't live together, or there is also a correlation. So the effects are different here. I'm not sure I understood the question.
No.
I think it's for simulated. You simulate that each individual has a different value, but as under the assumption that you don't know this value, you take the average and all the individuals get the same value for the estimation. For the value estimation.
And then
so that's what actually achieves Masoud testing. What is the problem being uncertain, whether the hair is too wide or not.
Yeah. So the problem, so this assumption is a worse assumption depending on each scenario.
Yes,
it's a better or a worse
assumption. The heterogeneity because 30% that means that one hair actually have 1/3 of all the possible scenarios you could have. So at the worst case would be when everybody have the same level, so everybody will have the same effect. So
Thank you.
Is there any question online? Can you still hear us? If you have a question, you can just unmute yourself. And I feel like I'm speaking in the void. And if you don't have a question, then we can maybe move on to the next presentation.
Okay. So here I used basically to do the analysis that you saw. I developed a set of tools that I use for that study, and also I'm now using it for another study. And this is basically a genomic selection simulator that allows for custom model. So I will start by recapping what I showed you earlier. So this is the workflow, very standard workflow for the genomic selection. You start by simulating a gene pool, simulate some biological traits. In my case, they were production potential and resilience. Then I had a random regression model that combined these two and made the realized performance of these individuals. So in this workflow, something I should say, the blue boxes are processes and the white rectangles are data. And then a genomic evaluation gave me the breeding values for each individual. I had different selection strategies implemented here to identify the next generation parents, and a mating strategy to simulate the next generation of offsprings. So this is a standard that all the pretty much what AlphaSim or QSim does, and what I also did. However, sometimes we need to change this random regression model. So for example, in the other analysis that I will tell you shortly, I need to completely change this random regression model to something very nonlinear. So to tell you a little bit more about that, I will start a little bit of detour. Currently, because we have a lot of automatic machines that can record the individual performance on a daily basis, these sensors and cameras, we can get pretty much the curve or like the trajectory or daily records of an individual on every day. And then we can observe and compare it against a potential target curve or target trajectory for that individual. And when the animal faces a challenge level, it deviates from that target by some value. I mean, it can die, situation happen that it can never catch up, or it may recover at some point and catches up to the target trajectory, which is the red line. Now, what is suggested in the literature recently is that you can basically take the deviation of the realized performance of the animal over time with the target and do some basically aggregate this data through some statistical tools like mean square variance, autocorrelation, and integral. And this gives you an indication whether the animal is resilient or not. For example, if I calculate these deviations and estimate the deviation and take the integral, I would expect an animal that is resilient to have high integral, has an autocorrelation that is around 0, skewness that is around 0, and so on. So these measurements are abstract, and people are suggesting that we should select for these traits. But the question is, if I select for integral, for example, or for autocorrelation, what exactly happens after 10 generations? What sort of animals will I have in terms of biology? So basically what happens is that these resilience indicators are mapping these longitudinal data into one value which I want to select for. So to answer these questions, what happens if I select for these and what sort of animals I get after X number of generations? I want to simulate this, which means I need to simulate this longitudinal data first before applying a resilience indicator on top of that. Fortunately, there are models that can simulate this longitudinal data. They are called mechanistical models. So they start by taking input some biological traits like the feed efficiency, growth potential, pathogen resistance, and so on. They also take out like how much pathogen the animal takes and work out an energy balance. So for example, say this much of energy goes for the maintenance of the individual, for the growth. Then also they couple the pathogen life cycle, as in like what happens to the pathogen, they reproduce and so on. And they give you the, for example, body weight over time, the black figure here, but also give you other information like the fecal count, the mass of the wool, and so on. So these models, basically, they map the biological traits to longitudinal data. Now, if we combine these two, what I get is a new workflow, which is now starting from simulating the biological traits. I go through a mechanistical model to get these longitudinal data, and then I go through a resilience indicator to get, for example, the integral. So that's it. I have made a new workflow which works fine, and now I can study what happens if I select, for example, for integral. But actually, if you look at this, this is like the previous picture that I showed you, except now that these three parts are together. So I can have a new generalized workflow where this random regression model that you saw earlier is replaced by the user's model. And the user's model is a function that maps this biological trait to some selection trait. So this has been developed in Fortran, all these blue boxes, but because Fortran is old and we don't want to force people to write Fortran function, I am now making an R package interface for this. And the user basically needs to write this F function, and some possible cases that we could use this. For example, in the previous study, I showed you how we can use production, like the biological traits here, or the production potential and basically a slope of the line. And we wanted to we analyze the realized performance of the individual. It can as well be used as in the classical study where we have genotype by environment interaction. We want to improve the performance of the animal in the bad environment, and we will see what happens, the performance of the animal in the good environment. In the other example that I just showed you, I have the output of resilience indicator integral. I am selecting for integral, and I want to see what happens on, for example, the growth potential of the individuals. Another possible case is where you have infection status of individuals for a disease. You select for individuals that are less infected, and you are interested to know what happens into the susceptibility of the host. And the potential use, again, is where you have your function here is a deep learning model, which basically predicts the imputed phenotypes from the genotype of the individual. So yeah, I mean, these are some of these possible uses that can be used in this workflow. So if you have any suggestions, or like you think this could be relevant or integrated into something else, or something is wrong, please give a shout. I'm also have some to-do list to do. So for example, in the initializing the gene pool back in 2019 when I started, we didn't plan to have a product like this. So we just wanted the gene pool for sheep. So I used the iterative method to basically evolve the sheep from a base theoretical population in Hardy-Weinberg equilibrium. But now there is coalescent method. I learned that it's direct, it's faster, and the people have written code for it. So one of the things I want to do is, instead of this iterative method, it just uses the library for the coalescent method, so it can make any gene pool for any species that the user wants. The biological trace, I'm assuming a standard, normally distributed effect of the QTL on a trait, but one can argue that not always the effects are normal, or then there might be a QTL with large effects. But at the moment, what I'm expecting is that the user assumes the effects are normally distributed, has a correlation structure, and however number of QTLs they think per chromosome affects the biological traits. The input also requires some genetic parameters like heritability, mean values, and variance components. The function here, again, F can be anything, can be an identity function. So you want to improve biological trait. You don't have any selection trait, and so this function here is an identity, or it can be as complex as I showed you in the earlier slides with multiple stages. As an input, it should get some traits for however number of individuals and some parameters, and outputs the selection trait that the user is interested in. So at the moment, I have implemented 3 genomic evaluation, like single trait random regression or 2-step reaction norm, but I don't want to compete with the BLOP-F19 or whatever. So at the end of the day, this should be an option. I have not implemented that the user can put whatever software they want to do the genomic evaluation. And then the last 2 steps, this can be, yeah, I mean, I have a few options for the user to do selection and mating strategies they want. But again, these are the simple stuff. The backbone is really the gene dropping and the selection that is done in Fortran. And yeah, thank you very much. If you have any feedback, question, suggestion on this tool, I'll be happy to hear.
Aside from the fact that everybody should look Fortran, in this example, you're showing us how to use the integral when we are dealing with a non-linear resilience. But how would you account for the stability of the system? So in other words, when using integrals, so in other words, you probably prefer an animal that follows a stable line rather than going down and up. So how would you account for that?
No, not really. So the integral, so this is something that is discussed in the literature, right? So you want animals that oscillate around the line to be resilient. So if you take the deviation and if you take the integral for animals that are very resilient is close to 0 because the negative part and the positive part cancel out.
Yeah.
So you will select for individuals that have higher integral. I mean, individuals cannot go over. If they go over the trajectory, you have estimated the target trajectory wrongly. Yeah. So yeah, it assumes that you know a target trajectory either by pre-infection data. So like have trimmed the part that is below here. So if you have the pre-infection data, then you can. That's a different story, like how to get the target trajectory.
You could select deviations from the trajectory.
Yeah.
So this refers to the alternative definition of resilience, right? Because that's what you hinted at and we divided resilience as a slope. And what do you do if this relationship between environmental challenge is not linear? So it's actually not a good measure, this linear slope. And so there's, I'm not sure how familiar you all are with the literature, but there's always, how do we estimate resilience? And one of the definition of resilience is a resilient animal is the one that doesn't deviate much from its target or quickly returns to it, right? So it's saying that you have a target, every individual has its target for producing in the optimal environment, and resilience is how much it deviates from it. So how long, and these are all these indicators that kind of capture that. And there's loads of studies that now are, as Masoud was saying, more data become available. There are loads of studies that say, yeah, we should select for those. And the question is, well, what kind of animal are you getting if you're selecting for those, right? Because it's so abstract. So one needs to look a little bit into that. And that's why maybe such a pipeline could be useful, because it lets you connect the biology with the abstract measures that you can select on. That's basically the philosophy behind it. But you said that in English.
I don't know if you are aware, there is a selection experiment for resilience in rabbits. What we did is it was resilience of litter size. So we didn't do any simulation, we did directly the selection experiment. And what we did is to compute the variance of litter size, longitudinal data of litter size, and we computed the variance of that data.
The variance of the raw data or the deviations?
So we first pre-corrected the litter size for systematic factors. Then we had the residuals of this litter size. Then we computed the variance of these residuals. So actually the selection criteria was the residual variance of litter size. Okay. And they selected now for 17 generations for that. And the animals, to biologically know what happened there, they measured the cortisol, and based on the cortisol, the animals were actually more resilient and also were more productive because they have, so those that were able to have homogeneous parities, they have also bigger parities always. So they were resilient and they were also more productive.
So these animals were exposed to some sort of challenge in their life?
No,
Okay.
the environmental challenge that a regular farm can have.
Okay, I assumed that.
No, they didn't pressure it.
Interesting.
So the animal that has lower variance
Have lower variance and bigger litters.
Okay, sounds good.
And it's a very long experiment. Rabbits has a very short generation interval, so in few years they will like almost 20 generations.
It's very, you're right, it's very, we don't know what is going to happen, but I saw that when you actually increase the mean or something, you get a scale effect and the variance is bigger.
Yeah, no, but
in this case, you actually say it's contrary, that it's like a coefficient variation that you decrease. Very interesting.
There is another one in this time with the variation in body weight of in mouse. And I think this one's got a different thing. Like it didn't happen that the more homogeneous were bigger. It happened that the more, I'm not sure about it. I have to check. But I remember that there was some inconsistency.
Interesting to know the citation.
Yeah, please, if you can, if they are.
Yeah.
Is there any question online? I'm trying to ask in the chat, but I don't see. I don't know if people can see the chat. If you have any question online, can you unmute yourself and ask it? Is there any question in the room? Final questions?
Perhaps a more simpler question. Why did you go through all this trouble of developing a new software where you already have so many softwares out there that you can simulate the population and play with parameters? Yeah,
so as I mentioned, I don't know, but can you go and change the model that, I mean, the structure is that you normally what you get is you simulate some traits, and then you are interested to improve some, or you select on a correlated trait based on the traits that you simulate. So the selection trait and biological traits are correlated, and they have a simple correlation structure. But what I have here is that this model can be as nonlinear as you want, and the correlation may change over time. And this is something that is not available. So by having a custom function here, basically the user model here, you allow the user to put however function they want. Yeah, and I'm hoping this will be used somewhere, as in in some of these cases could be used or more.
And then another one, so you showed different ways of measuring resilience or skewness, whatever.
Oh yeah, so this is not my work. This is it has been suggested that you can, for example, as Mariana suggested, that you can select on variance or on autocorrelation.
Have you considered a study that would assess all of these sort of?
I'm now doing a simulation study on some of these to see if you select on, because these the mechanistical models are in the literature. Everything is on the literature. The tools are there, but a simulation study has not been done to see what happens if you select 10 generations on autocorrelation. So this is what I'm doing at the moment.
Can thank Masoud again. Thank you. And I'll stop recording. Thank you for attending here in person and also online. And see you in the next seminar.
Okay.